to last between 30 to 45 minutes just to cover the last part of viruses. So if there is no questions or anything like that, I'm just gonna jump directly to the lecture. If anyone has any question or something, throughout the class, just raise your hand or interrupt me and ask me so we can try to figure out the best answer to your question. All right, so let me go back to sharing my presentation over here. And just as a quick recap, and for those ones who weren't here the last time, just quickly, we're gonna review again the replication of bacteria. So remember, I'm trying to say replication and not reproduction, because replication implies something that is not really alive. Reproduction is like for life forms, and these structures or viruses are not really alive. If you compare it with their characteristics, which are over here, they actually do not have all the characteristics of life. They don't use water. They have to use other cells in order to replicate. So they don't reproduce on their own. They don't have any, they don't have any stimulus to the environment. They don't react to the environment that way. However, since they are genetic material, they are subject to the rules of evolution and natural selection, but besides that, all of the other characteristics they don't possess. So don't forget, absolutely all of the viruses are obligate intracellular parasites. That means that they have to go inside of a cell and they have to use the machinery of the cell in order to replicate more copies. And usually those copies can go from one virus infecting one cell is going to produce at least 50 viruses to thousands of them. So once they're ready to leave and they're assembled inside of the cell like a factory, like they burst the cell and then they leave. So even they make their own cell burst. So there is an enzyme I think is called lysosome. I'm not sure. I have to review it again, but actually that enzyme is capable of breaking the cell membranes of cells. So the own cell is going to create their own death. So they burst and then they leave. So let's review quickly the replication of viruses. So we are going to see it again over here step by step. So the first one, don't forget, is going to be the attachment. So attachment is going to be very specific to the tissue and the organism that the cell is going to be infected. It's going to be infecting because the viruses are going to contain some specific compounds and proteins that are going to be over here, and they're going to function as a Velcro with the cell membrane of the cell that is going to get infected. And once they are attached, then there's going to be a section part, which is going to be called penetration, in which the virus is going to insert their DNA or RNA inside of the host cell. So the third step, and I unified this one, is going to be replication slash synthesis, which is this one over here. Number 3 is what's going to happen in this one is like, say, the DNA of the organism is going to be here. So this section of DNA or the virus is going to get inserted over here. So what's going to happen is like this section of the viral DNA, what it's going to do is going to dictate now to the cell to make more of the pieces of genetic material, and then the capsule, the envelope, and all the pieces of the viruses. So replication slash synthesis, that's when it want to happen. On the fourth step, which is this slide over here, is called maturation slash assembly, is when all, like in the factory that I was mentioning, all the pieces of the virus are going to get together and start getting the shape of the viruses. And then on the fifth step, which is release, the cell is going to rupture, and then it's going to release all the mature viruses ready to infect again. So if you think about COVID-19, if you think about the influenza or the flu, if you think about Ebola virus or the cold viruses, and those ones, they exactly follow all these processes over here. Just a tiny difference, it is Just let me jump over here. With the case of animal cells, in the case of when they're infecting a cell like they do with us, in the case, for example, of coronavirus, what's going to happen instead of injecting the DNA or RNA over here, what they're going to do is like the whole virus is going to penetrate the cell. And once it is inside, it's going to release all the components and the material over here, the genetic material. Don't forget the genetic material is either DNA or RNA. If it is a DNA virus, the DNA is going to travel to the nucleus and then is going to insert into the DNA of the host cell. If it is an RNA, it can go directly to the ribosomes because remember the ribosomes are going to be the ones making proteins, and then they're going to produce everything over there. And then all the assembly and then the release is going to happen. And in many cases, like HIV viruses and other type of viruses, what they do is like when they leave the cell, they actually don't kill it right away. What's going to happen is like they're going to take a coating of or a piece of the cell membrane of the host cell, and then they're going to be hiding like the wolf covered with a sheep skin. So it's going to be hiding. So when the immune system is trying to recognize if this is a virus or not, they're going to think that it's a common cell and they don't need to attack it. So that's why viruses keep replicating and replicating and replicating without our immune system recognizing that they are antigens or things that don't belong to our bodies, so they won't attack them, okay? A little bit of the classification, this is where we left off last time. So usually viruses have been classified depending on what organisms they infect and the type of tissue that they attack. If they can be like dermotropic, if they attack the skin, neurotropic, if they attack the nervous system, viscerotropic, if they affect like all the stomach, intestines, and related organs, pneumotropic, like the coronavirus, that they attack the respiratory system. But actually, like there is this committee, which is called the International Committee on Taxonomy of Viruses. They join every 4 years and they try to discuss newer classification for this. But the fact is like, since they are not a real organism, they are not species, it is more difficult to classify them. So of course we can classify them by similar characteristics, by genetic material, by similarities between each other, but still is kind of difficult to classify them. So the easiest way that the ICTV has determined how to classify these viruses is either if they are RNA viruses or if they are DNA viruses. So if you notice, there is nothing phylogenetic over here. There is no evolutionary relationships between these ones who was first viruses. There is an S missing over here. If they are these ones or these ones, actually, like the ICTV just leaves this like, okay, let's classify them by the type of genetic material that they have. Don't forget again, viruses, they contain only one type of genetic material, either DNA or RNA, right? So here are some examples that I'm going to tell you about RNA viruses and DNA viruses. So most likely what I'm going to do for the exam, I'm gonna write a note, like a kind of a study guide quick for a reminder for even the people who weren't here. I'm going just to write, say, the names of the families during the exam, some of the names, and I'm gonna tell you, are they RNA viruses or are they DNA viruses? But since you are going to have these slides over there, you're gonna be able to go back to the slides and check and say, okay, so RNA viruses, so for example, like the picorna virus. If you heard about rhinoviruses before, anyone heard about rhinoviruses? It's not a virus that is affecting the rhinoceros in Africa or anything like that, but actually the rhinoviruses, we have like over 100 types of rhinoviruses, and they actually are the ones that they produce the common cold. So one of the main reasons that we don't have a vaccine for a common cold is because we have over 100 types, over 100 types of varieties of rhinoviruses. So imagine trying to get a vaccine that is going to cover 100 different types of viruses. If we barely made one for COVID-19, which we have the whole genome and everything, it's like imagine trying to group 100 different types of these ones and group them together to be get immunity. So this is something that is not going to happen anytime soon. So that's why we don't have a vaccine for the common cold. Don't forget the differences for common cold and flu are slightly different. Usually the common cold is going to be faster and is going to act on the upper respiratory system. So what's going to happen mostly with the common cold is like your nose is gonna be stuffed. Then you're gonna have a sore throat really fast. You're gonna start sneezing even more, and stuff like that. And usually the rhinoviruses they follow a lytic cycle. That means that they are really fast. They follow only one cycle of replication, and they burst and they leave. So that's why the last time that I had a cold, I remember I was like, oh yeah, I'm teaching right now. And by 7 o'clock or 8 o'clock, I was feeling really bad. I had fever and everything because they were really fast acting. And usually they last 3 days, 4 days, and that's it. With the differences with the flu viruses or influenza viruses is that actually they infect even the lower respiratory system, so they can create even coughing, and they're going to be a little bit slower acting, and they're gonna last longer in your system. So a flu might last 6 days or something like that, compared to the no, no.
Hello, guys.
Back.
Give me a second, hold. Okay, sorry about it. I hate my internet. I was telling you I hate my internet. Okay, so let's continue with this. I'm sorry for this. So where what I was saying, I think I was saying about yeah. Okay, thank you everyone. Can you hear me? Yes? Yeah, all right, thank you. Okay, so as I was mentioning before, viruses. So I was talking about rhinoviruses, the common cold, but also we have other RNA viruses like hepatoviruses. They produce the hepatitis A. So actually, like now we were talking about diseases, you'll see that because I lived in Guatemala for like 20, 21 years. Well, every time I mention parasites, I'm gonna mention that I had those parasites because actually, like Guatemala City, they have water treatment plants that are really bad. So when I was a kid, I actually I had hepatitis A. So hepatitis A produces an infection on the liver. So I have inflammation on the liver. So I had like my eyes yellow and my skin was a little bit of yellowish. And I remember I was really happy even I had the disease, but it is because the doctors say like you have to eat a lot of candies and everything and sweets when you have this type of diseases like hepatitis, because in the on the liver we have some reserves of sugar. So since the viruses are destroying everything, then we don't have those reserves of sugar called glucogen that are going to be used in case that we have a drop in sugar blood in our bodies. So I was happy, and I remember I was eating Snickers, I remember I was eating lollipops, and I was eating all those things that I wanted, but I was like 7 years old, so I cannot remember that. Enteroviruses, if you hear about the polio virus, one of the presidents of the United States, they had polio virus. So they usually invade the intestines and everything, but later they can infect the nervous system and they can cause paralysis. So that's why sometimes when you don't get a vaccine of polio virus, then a child that can get infected, then is going to have assistance for walking because then the nervous system is going to get affected. Other types are going to be flaviviridae, for example, hepatitis C. So hepatitis C is deadlier than hepatitis A. So hepatitis C, you see there are belong even in different type of groups of RNA viruses. If you hear about mumps, measles, and I'm pretty sure you got a vaccine, it's called the MMR, which is the mumps, measles, and rubella. So this vaccine actually is focused into this group over here, and then it's going to create immunity in you once you get the vaccine. If you heard, there's a chat, somebody's telling me, do we know the families and their definitions? So actually, you don't need to know the families and their definitions, but what I'm going to do for the exam is like I'm going to put, based on these slides, I'm going to put, okay, the following viruses are RNA, and then I can put flaviviridae, paraxomiviridae, and picornaviridae. And then you're gonna be able to go back to the slides and find these viruses and be like, okay, yes, this one, yes. But maybe I can put like picornavirus, paramyxovirus, and let's see, hepatiniviridis. So you're gonna be like RNA, RNA, and this one is DNA. Okay, so this option is not the one that I need. Yeah, you're gonna make you look a little bit more into the slides over here. So you don't need to remember this. Trust me, like this is a lot of information. So I'm just giving you an example of all the diversity of viruses that we have. So let's see where I was over here. So if you heard before rabies before, like have you had it or you heard of any dog that had rabies before, and then they have, or they also called it hydrophobia. It's like, and they have a lot of like white foam in the mouth and they get really like aggressive and the behavior changes and everything. So actually that happens because these rhabdoviridae, the viruses for rabies, actually they infect the nervous system. And what they do is like there are two options with rhabdoviridae or rabies. It's either the animals, including humans, they can get really lethargic. Or number two, they can become really, really aggressive and violent. Even say if you have a dog and you've been having your dog for 10, 12 years, and you think your dog wouldn't do anything to you, and if they get infected with rabies, there might be a probability they can even attack you. And they have a lot of foam in the mouth because they call it hydrophobia. So phobia means fear and hydro means water. And the problem is like the throat is so closed that actually they cannot swallow anything, not even the saliva. So that's why the saliva looks like foamy, because animals, they can't even drink anything. So that's why they call it afraid of the water. It's just because they just can't swallow water. So that's why, like, if you watch videos of rabies, animals with rabies, you're gonna see like they have this characteristic of foam in their mouth because of that. And before, I'm not sure up to now, but anyone knows the type of vaccines and how this vaccine was administered before for the rabies?
Didn't you used to get 21 shots in the stomach?
Yeah, exactly. So you get like between 14 to 21 shots, I mean, injections in your belly button. So, or in the stomach area. So mine, you have to be receiving once every week, because this was, because if you don't treat rabies, you're going to die. That's something certain. So that's why many animals and everything, when they get rabies and they bite another animal, then the best humane thing to do, they say, is like better sacrifice them, because otherwise they can damage other animals, so they can suffer a lot. So we have other viruses over here. These ones especially, these ones are mainly in some tropical forest of Africa. And many of these ones, like the Ebola, is thought to be present in, say, I think gorillas or chimpanzees, but due to the pressures that we have for hunting and eating them and everything, these many viruses, like they jump to humans. And Ebola actually is a filamentous um, virus that actually what it does is going to affect your red blood cells, and then it's going to start destroying everything, all your red blood cells, and then you start like dying of like leaking blood all over the place. So this is a really bad death, and they say like the probabilities of dying of this once you contract it are really, really high. So let's just forget about this one, crossing fingers that we won't have a pandemic with this type of viruses, because these ones are going to be really terrible. If you've seen those sci-fi movies with a post-apocalypse virus, I'm pretty sure it could be one of these ones over here, the hemorrhagic fevers and everything. Reoviridae is, I want to skip this one over here because I wanted to talk more about the retroviridae. So retroviruses, the beauty of retroviruses is like they have the RNA. So, you know, like when you're speaking about the general movement of information, so RNA is supposed to go to proteins, right? Into the process of translation. So translation over here, so what's gonna happen is RNA is supposed to move to proteins. But what happened with these retroviruses is like they call retro because once they are inside of the cell, they're gonna go back into transcription and then they're going to make viral DNA. So once based on this DNA, then they're gonna go jump back into RNA and then back to protein. So that makes it more difficult for medication to work because they're working backwards. And as it says over here, and usually many of these ones, they can cause tumors and leukemia. And many of these ones, and especially one that there is a pandemic actually, like a silent pandemic that is happening since the 80s, they can infect immune systems or called the T lymphocytes. These ones are the ones in charge of noticing any disease or anything foreign in your body, and they're going to detect it, and then they're going to raise a red flag and say, hey, this is a disease, come and kill it. So what's gonna happen, like these viruses are going to kill these ones, so then our immune system is going to be so weakened that we are unable to respond to infections. Even to the minimal infection that we can have, then we are not gonna be able to respond. And then we are going to acquire something called the human immunodeficiency viruses. So if you heard about the HIV, so this is the type of viruses in which HIV stays. So this is the HIV, the one that causes AIDS. Anyone heard about AIDS before? I'm pretty sure everyone heard about the acquired immunodeficiency syndrome, right? So this is actually a pandemic that has been happening since the 80s, and it's very difficult with HIV also to track them, not only because they go back to DNA, but also it's like when they leave the cell of the host, they're going to take a coating of the membrane, so then they are not really recognized for anything. So people actually don't die because of the virus, is because people don't have more lymphocytes that are essential to flag an infection, and then the immune system is never going to react. And then if you have any simple infection, then you're going to die. So that's a problem with this one. Sometimes there are treatments in which there are transfusion of like white blood cells, etc., but still we don't have a cure for these type of viruses up to now. So hopefully soon we're gonna get a cure for this one. So this is what I was saying. It takes a coding over here. So this is a video that you can watch later. Then coronaviruses, wow, like something that we have right now. So they are RNA enveloped viruses. The envelope has peplomers. So if you heard about the spikes that everyone was mentioning in the news and everything, they're going to function as a Velcro when they get attached to the respiratory cells that we have in our system. And then usually coronaviruses are going to cause diseases in birds and animals. Most of the time, as it says over here, is going to cause respiratory illnesses, including sometimes the common cold, although having the symptoms of common cold is going to be very, very, very rare. Many of these coronaviruses are going to be infecting other animals, as I was saying, birds and mammals, and very few ones are going to be infecting humans, like the severe acute respiratory syndrome of SARS that we had in 2000, I don't remember the year, 2009, 10, something like that. They have the Middle East respiratory syndrome, and now we have the severe acute respiratory syndrome coronavirus 2, so version 2. So version 1 was the first one, and then this is the one, aka COVID-19. COVID for coronavirus, and then 19 because of the year. But it is known that these ones are zoonotic diseases. That means that it's coming from an animal, and due to a mutation is going to jump into humans. And now the humans are going to become the host of these viruses. So that's what it happened with zoonotic. And that's what it happens when there is a need of, say, of more land, of people are cutting down trees because they need to have more land for farming, etc., etc. So some of these animals that are wild getting close contact with humans, and then sometimes humans even eat them because the need of eating protein, you know, and then these diseases for a rare mutation by mere chance are going to mutate and jump into humans and infect us. DNA viruses, the adenoviruses are actually the cause number one, almost cause number two of gastrointestinal infections. So usually it's like Salmonella, Listeria, and adenoviruses. So usually when you eat something and then you feel sick 2, 3, 4 hours later, it is most likely that you have an adenovirus, because usually bacteria that you you have food that is contaminated with bacteria is going to take between 24 to 72 hours to develop. When you eat something and you have an intestinal infection, it's most likely to be a virus infecting you. Then we have herpes viride. If you hear about the smallpox, so people who was born before 1979, all of them, they have been vaccinated by smallpox. The ones that we were born after 1979, we are not vaccinated with the smallpox vaccine, so we are not immune to smallpox. So it is known that we have, in the world, there are 3 laboratories that they contain smallpox still kept over there in the laboratories. So 2 laboratories are in the United States and 1 laboratory is in Russia. So they still keep it, they say, for research purposes, but many other people think that maybe it is because in the case of a warfare and the other country is throwing like dirty bombs or like biowarfare, then we still can develop vaccines and then give the vaccines to people to get immunity. Otherwise, this disease is really, really lethal. So everyone born after 1979, we don't have this immunity for this. More viruses over here. If you care about the papilloma virus, is in this one. Hepatinovirus is in one hepatitis B. So remember, hepatitis A and C are RNA, and hepatitis B is on a DNA virus. Then we have other types of viruses that I'm just gonna jump over here. So regarding culturing viruses, so you know, like you cannot just have a Petri dish with a gelatin over here, like with a lot of nutrients, have the viruses start growing over here. Because we cannot do that, because actually viruses, they need something alive to grow. So what they used to do before in the 1930s is like they were using embryos of chickens. So people were saying, yeah, let's just have a lot of chicken embryos, and they just grow viruses over there. So when they were still on the egg, they were injecting them with a tiny needle and having them grow over there. But scientists were saying, you know, this is kind of like, we need to develop bioethics and not to use living or other living organisms for these purposes. So what now we do is like we have something called the tissue cultures, in which in a Petri dish, actually, we have the specific nutrients so we can grow cells. And then in these cells are growing in the Petri dish, then we can produce viruses from there. And then from these ones over here, we have a bunch of viruses that we can later take from there. So say for culturing COVID-19, probably they can swab the epithelial parts of the nose or part of the throat or near the lungs, the bronchioles or something, and then they can make them grow by mitosis in the Petri dish, and then they can inoculate them with COVID-19, and then they can make COVID-19 grow over there. So instead of using chicken embryos or anything like that, it is better to use this type of techniques. So let's go quickly to the part of antivirals. So this is the last couple 15, 20 minutes that I'm going to use. So there are two differences, antivirals and vaccines. So the antivirals used to make the difference, and antivirals are not going to destroy the target virus, but what they're going to do is just like they will inhibit their development. So what's going to happen is like, say you have coronavirus with the spikes outside. So what the antiviral is going to do is like it's going to attach over here and attach over here and attach over here. So actually the virus, when it's trying to attach to the host cell, is not going to be able to attach at all. So that's what is a very simple way to put it. That's what can inhibit the encoding or the entry of the shape of the viruses. So even if this one enters, then it's going to enable to break down. So then the virus is just going to be inside, but then it's not going to replicate. So for example, the MMR vaccine is going to be an antiviral because what it's just going to do is just going to have this. So if you got vaccinated by the MMR at some point, and I'm pretty sure all of us we did, we have these antivirals that are constantly in your blood that are going to be there. And if one of these diseases, measles or mumps or rubella, are going to infect us, actually we have the chemicals inside of us, I mean the parts, the components that are not going to allow this virus to infect us, right? On the other side, we have the vaccines that they were developed about in 1796. You see, like a long time ago by this scientist called Edward Jenner, who was actually studying cowpox. So the cowpox is similar to the smallpox. So what he was doing, it was like he was administrating, what he was doing is like heating up a solution of plasma of cow. He was doing that, and then he was like killing the virus. He didn't even know what it was, but he was like technically with heat, he was killing the virus, and then he was injecting it to people. So the first attempt that he did, I believe, is like he was injecting a kid with this one, and then he exposed it to the smallpox. You see, like before they didn't have like ethics of working under certain conditions. So he was just like, I'll take it, I'm gonna inject you with this, I'm gonna give you this medicine, and then I'm gonna expose you to smallpox. And he noticed that actually the kid didn't get infected. So he was like, okay, so this is something that might work. And then we give it the name of vaccines because, well, the terminology in Latin, and I don't know the very well in Latin, but I know like for those ones to speak any Romance language like Italian, Portuguese, Spanish, you know, vaca means cow. So that's why it is a derivative because it's coming from cows. So the very first vaccine is coming from cow, which is like vaca in Spanish and other Romance languages like that. So that's why it's coming like this word. So how effective is vaccination? I'm just showing you over here the cases of diphtheria, pertussis, and measles. So if you get the vaccine, the Tdap, so if you hear about all those vaccines, if you see the cases before, this is before and this is after, this is before and this is after, and this is before and this is after. If you can see, the statistics are clearly self-explanatory, in which right after the introduction of those vaccines, we almost don't have cases of anything right after this. So sometimes we have like probably up and downs over here, but it's nothing compare like say pertussis cases over here versus these ones over here. Not even not only for this one, for measles, so we had like a lot of cases, but then after the introduction of the vaccine, like we have even less. So probably this one because people were accepting the use of vaccines. So that's why you see a reduction over here of the cases of these diseases over here. So how do they work? So what's gonna happen with vaccines is like we have two different types of immune system. First of all, we have, first of all, we have physical barriers. Like, you know, we have the mucus, or the, if you call it, the buggers in their nose. So those ones are going to try to capture everything, and sometimes that's why they're like sticky or something. That they want to try to capture dust particles, viruses, bacteria, and everything. The skin is going to be another barrier against diseases. But if they go through that, then we're going to have something called the innate immune system. And the other one is going to be called the acquired immune system. So vaccines are going to enhance, are going to improve or acquired immune system. It is acquired because then we are teaching the immune system how to react. So for the innate immune system, we have specialized cells that are going to be just like eating. Technically, there's a type of cell that we have is it's going to be eating material that doesn't belong to the body. But then we have the acquired one, which is going to learn how to fight something. So that's why we get, once we get the vaccines, we are going to produce something called antibodies. And then these antibodies, so usually they have a white shape. They are going to be present in your body for a specific amount of time. So actually, even I heard that we might have to take another shot of vaccine of COVID-19 at the end of the year. So they're still investigating how long these antibodies are going to last, because some antibodies like the measles or the, for example, for those one with the for the vaccine of smallpox, they might last your whole life, but some other antibodies are going to last probably 2-3 months. So for example, the antibodies for the common cold, since there are too many, they're gonna last 2-3 months, and you can get sick again of the same disease. And the same with the flu and everything, because they're going to last just a few. So David, I have been told, so I'm just reading what David is writing. I have been told that viruses cannot be cured. So yes, actually, once you are infected, then the only thing that you can have is treatment just to pass the disease. But this is prevention. So usually the best medicine that works is called prophylaxis, which is just like prevention. So this is just a way to prevent the disease. Usually some antivirals, they're going to help you just to, as I was mentioning, just to pass the disease, because you are not going to have more viruses to be growing or replicating inside of you. But still, the damage is done, so you only have to will survive that infection. So vaccines are going to be to prevent. So it's not for curing, it's for prevention only. So thank you for that. That's a really important key over there. So as I was mentioning before, the antibodies are going to be in your body. So when the disease is going to try to attack you, actually, you're going to have these antibodies that are going to attach to the, say, in this case, the COVID-19 virus, and then they're going to be like a red flag for different immune system cells, like they call it cytotoxic cells, the K killer cells. So what they're gonna do is, like, when they recognize this attachment between antibodies and viruses or any other pathogen, they're gonna come and they're going to destroy it. So destroy it, or either gonna engulf it, and then they're gonna eat them and then destroy it on the inside. So that's what vaccine is going to do, is it's going to enhance, is going to improve or acquired immune system. So again, this is just another picture of what I put over there, how vaccine works. Again, the antibodies are very, very, very essential. Remember, for any pathogen or anything that doesn't belong to the body, we're gonna call it antigen. And I remember we spoke a long time ago for when we were talking about proteins in the biomolecules, it was antigens. So antigens and antibodies. So antibodies are your friends, and antigens are going to be our enemies, okay? Types of COVID vaccine, the ones that we are actually using now is the DNA RNA, which is the most elegant way now to do vaccines, because what is happening, how they do it, is like they took the sequence of the RNA of the virus, and then they were studying what sections, which genes were the ones that were creating the virulence of the virus. And they were like, okay, so this gene and these are the ones that actually are causing the damage. What we're going to do is like, we're going to then have like reverse engineer, have the body to make antibodies for this. So we are going to design a specific RNA that is going to be the antivirus over here. So this RNA, once is going to get injected as a vaccine in your shoulders, like many of us did already. So this RNA is going to go to our cells, and then it's going to be inside, and then it's going to go to the ribosomes that we have, and then through the process of translation is going to produce a bunch of antibodies that are going to be on the blood, constantly looking if we are infected or not. If we get infected, then they're going to get attached to the COVID-19, and then other cells of the immune system are going to come and destroy. So this is what happens over here. So technically, the vaccines that we are getting, the mRNA vaccine, that's why I call it messenger RNA, is telling us that we have to do these specific antibodies to kill COVID-19, all right? So that's what is happening right now. It's really elegant because before, the other types of vaccines that we had is like these ones over here, is mostly that we heat up the viruses, so we destroy some of the components of the viruses, or we eliminate the DNA or RNA, or we take only pieces like this one subunit, and then we produce the vaccine like this one. So usually these ones are also really, or these ones are like the classic type of vaccines, but like the ones that we are having right now for Pfizer and Moderna, I'm not sure for the Johnson & Johnson, but Pfizer and Moderna, is this reverse engineer that scientists are using for this. Okay, what else? So anti-vaxxers, anyone heard about anti-vaxxers before? Is anyone an anti-vaxxer over here? Raise your hand. Nobody? I see Adam saying yes. I see Lisette kind of smiling over there. So like, anyone can tell me what is an anti-vaxxer or what is an anti-vax movement?
I'm not saying I'm an anti-vaxxer, but I think an anti-vaxxer is just someone who doesn't want to get a vaccine or just doesn't believe in vaccines.
Mm-hmm.
Right.
Yeah. Yeah. No, no, no. And yes, that's correct. I mean, I also reading Adam's comment over here, like he's saying, wait, I'm not an anti-vax. No, I know. You're saying yes, like you heard about anti-vaxxers. But yeah, so anti-vaxxers is that person that doesn't believe in vaccines, and actually they believe in other natural ways of healing or something like that. So they think that actually vaccines are the ones that produce autism in kids or something like that. So this is the main thing that they were saying back in the late 1990s. There was a publication about it. I think the scientist who was saying that published a paper saying like, yeah, there is a direct correlation between vaccines and autism in kids. So many people were like, what the hell? I don't want my kids to be autists. I don't want my kids to be like this, etc., etc. So the movement began to say like, we don't believe in vaccines at all. So there was kind of like a conspiracy theory, even despite the fact that we have so much theories, so many vaccines and the statistics that they have shown the effectiveness of vaccines. So actually, even years later, the same scientist, the same person who published this paper, initiated everything, he discredited the publication years later, saying like it was just like fake information, and he was just like playing around with everything. So then I don't know what happened with him, but I'm pretty sure he's pretty well hated among the medical community. Also, there are some other facts, for example, like when they were saying like, oh, kids, they're having too many vaccines because their immune system is not able to withstand anything. So theoretically, like on theory, but it's not like, hasn't been applied because that would be too intense. But even newborns, like babies, they can actually respond up to 10,000 vaccines that can be given at the same time. That means like our immune system is so specialized and it can absorb all that information and create so many antibodies at the same time that you can even give like 10,000 vaccines of a baby in a span of 12 months. And then this baby, even a baby, imagine, like is going to be able to react and produce the antibodies for these 10,000 different vaccines. So these anti-vaxxers, I don't know what they were talking about. So they also were saying like natural immunity is better than vaccine required medicine. Maybe later I'm gonna send you a link in which there is a case in Canada in which these two, there was a couple that were anti-vaxxers. They had a kid who had meningitis. Meningitis is an inflammation of one of the layers of the brain. So one of the friends, she was a nurse and she noticed that and she told them to bring them to the hospital. And then they said, no, because we don't believe in vaccines and blah, 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 blah. So they gave him like a chamomile tea with organic honey and something else. And actually the kid passed away. So then they were charged with murder in Canada, and I think they were convicted because of they were neglecting health to the kid, and the kid died because of the really bad decision that they had. So the people say like natural immunity is better than vaccine acquired, and sometimes acquiring the disease normally creates a stronger immune system. But imagine like being exposed to the live virus might be deadly. Say I had fever for 1 day when I got the second shot of COVID-19. But trust me, I would prefer to have 1 day of fever versus one of my friends in Guatemala. She was with fever for 10 days. So she was feverish for COVID-19 for 10 days. So I would prefer to be like that. Or if you had smallpox, even the chances of acquiring immunity and chances of surviving are very, very, very low. So yeah, I'm gonna try. Yeah.
Is this the same thing that back in, like in the 60s and 70s, where parents used to, like somebody down the street would get chicken pox or measles and they'd say, go play with your friend so you can get it?
They still do it in Guatemala, you know. It's not even the 60s. They still, I'll go and play over there because you can get the disease over there. But yeah, people still do it in some places in the world and everything. And some of those diseases are not deadly now because even if you can get really bad, you can go to the hospital and they can treat you, antivirals and everything. But the thing is like there are other diseases that actually might kill you, like this meningitis that I was telling you with this kid. It's like it would be better to have a vaccine and suffer a little bit of dizziness or fever than suffering the consequences of that. So with measles and mumps, you can get scars. You will be like with the scars all over the place forever. So it's like, okay, I survived, but at what cost? I like have all scars and then something. Or with polio, yeah, I survived and everything, but then I need assistance to be walking because my legs can't move anymore. So it's like, well, it's better a vaccine, and sometimes you don't even have the side effects at all. So I'm gonna try to find that article and then we'll try to share it with you so you can read it over there. What else do we have over here? So almost, yeah. Yeah, vaccines contain unsafe toxins. Sometimes they can have like some elements like formaldehyde of aluminum that are used to improve the attachment or the diffusion of the vaccine in your body, but actually they're all of them approved by the FDA and the CDC. And even now for the COVID-19, they say like, yeah, some of those ingredients, some people can be allergic. So they have a warning that they should know the ingredients of these vaccines before taking the shot. So before we finish this, just quickly, so we thought that viruses were the smaller particles that were infecting organisms, but what if I tell you that it's not really, really true? And there are even some smaller particles that they can create diseases in animals and plants. So we have two types of other even smaller particles that we're using to mention really quick in these 3-4 minutes. One is called viroids. Viroids are going to be just RNA strands. So remember, a virus is going to be the RNA or DNA with a capsid, and then sometimes an envelope. In this case, a viroid is just going to be a single strand of RNA. That's it. So what it's going to do is, like, when it goes inside of the host cell, is going to produce more RNA copies and more RNA and more RNA and more RNA. So it's going to create a domino effect inside of the cells of the organisms, and then in general in the whole organism. And it's going to create, say, in the case of for humans, it's going to create like losses, because I don't have the pictures over here. I don't know what happened with the other slide. But usually the potatoes are going to look like having a lot of tumors. The growth of the potatoes is going to be really ugly. And the tomatoes are going to be looking with a tomato with a lot of bumps all over the place. So people wouldn't buy those ones, because actually they are also containing viroids, which is just RNA like this one. On the other case, we have prions. Prions are really, actually, if I would study virology, I would dedicate my life in studying these ones, because these ones is not even genetic material. Prions are only a protein. So they are proteinaceous infectious particles. It's just like a protein. You remember they have the tertiary, quaternary structure, and they are just floating in the air. And then what's gonna happen is, like, they're going to land in organism. Then they're going, for some reason, by just randomness, they're going to penetrate, go inside of the organism, and they are also going to create a domino effect in which they're going to unfold other proteins inside. So more and more and more and more. And sometimes the diseases due to prions are going to be seen, like what I call the symptoms of infection with prions might happen 20 or 30 years after you got infected. So as I'm saying over here, like, our misfolded row form of a normal protein is just going to be a different shape. And this one is going to cause a chain reaction between the proteins of your body. So we have, the picture of a normal brain over here, and you see the picture of a brain with a disease. So you see it looks a little bit like spongy. So actually, when you go and take a slide and put it under the microscope, it's going to look like these are normal tissue. But when you see the other one, it's going to be looking all expanded and like a sponge. So actually, we have a disease. We have several diseases. So the German, Straussler, Schneider, I don't know, somebody speak German, you can tell me. But this one is going to include slowness of thoughts, dementia, other muscular tendon problems, and the fatal familial insomnia. Can you imagine dying because you cannot even sleep? So it mess up so much your brain that you can't even sleep. So then that's gonna happen with these diseases. There is one that is called the transmissible spongiform encephalopathy, or TSE. What's gonna happen with this one is gonna happen this. It's going to create like a spongy tissue. Instead of a normal tissue, it's going to create a spongy tissue. And if you heard about the mad cow disease before, anyone remembers about the mad cow disease? So actually the mad cow disease, even there was a big case in England that some of the cows were having issues of behavior, and they were saying mad cow because they were just having erratic movements, and they were moving back and forth and roaming around, wandering around and everything. It's just because the brain was getting infected by these proteinaceous particles that actually they couldn't even move. So they had to sacrifice thousands of them because they were just like, they were done. So there was a chance, and many probably people who ate one of these ones, you never know, like 30 years from now, probably in 2040, 2030, some people might show some symptoms of this. Is because the proteins are little by little getting a domino effect. So I'm going to leave these ones over here, things to remember. I'm gonna send either way an email with the things to remember for this class. These are the main points. I was just writing them this past half an hour, but I'm gonna copy and paste and send an email to you with the things to remember to study over here. So is there any questions so far about it? Sorry, I said 45 minutes, we was 56. Viruses, scary, right? So we don't even know that they're around. Viruses, prions, viroids. So, now you know it's a little bit more about all that information. So when you're talking about COVID-19 with somebody, you can shoot like, oh yeah, RNA viruses, oh yeah, the spike proteins and lytic replication, and they'll be like, oh my God, how do you know so much? And then you'll be like, yeah, because I study biology. So any questions or anything? No questions? All right, so if there are no more questions, then have a good evening. Don't forget to finish your worksheet. That's the only thing that we need to do for this week. And then I will see you next week speaking about protists and fungi, okay? All right, thank you, everyone.
Professor.
Thank you.
Have a good night.
You too, thank you.